we mustn't forget that cardiovascular disease has sadly not gone away. And we still have a major problem to tackle. In fact, I would argue that the need for new discoveries has never been greater, with the quite strong evidence now that individuals with cardiovascular disease are at greater risk of severe outcomes from COVID-19, and that the virus may actually cause direct damage to our hearts and blood vessels and trigger acute events. And that's not even mentioning possible long-term effects of the coronavirus on the cardiovascular system, which we know so little about at the moment. And I know Sanjay is going to tell us a little bit about that later. So what's the answer? At the BHF, again, thanks to your support, we strongly believe that identifying and funding innovative research by the brightest and the best, wherever they may be, is the answer and is the way forward towards achieving our mission towards a world that's free from the fear of heart and circulatory diseases. It may surprise you to know that the British Heart Foundation actually funds 55% of all non-commercial research into cardiovascular diseases in the United Kingdom. As in fact, across Europe, it's the second largest funder of cardiovascular research behind the European Union. We fund projects and programs from fundamental science using cells and genes and molecules through to research in patients, including clinical trials. We contribute to major infrastructure projects, buildings and equipment. And wherever possible, to get maximum value out of the pounds that you entrust in us, we partner with others to move the field forward and to empower new discoveries through both national and international partnerships. But perhaps the biggest and the most important investment we make is the investment that we make in people. From researchers at the pinnacle of their careers, such as our BHF professors and senior fellows, to younger researchers, the leaders of tomorrow, like our PhD students. Now, I don't want to just emphasize the quantity and breadth of the research that the BHF funds. I think it's also important to emphasize the quality of the research that's enabled by your support. So I'm just going to show this, this chart. It may look a bit scientific. The main point, I guess, is that the taller the bar, the better. What this shows is the influence and impact of research publications arising from work funded by different funders. This is an independent analysis very recently published by the Wellcome Trust. And the second point I'd like to point out, and you've noticed that already, the BHF is in red here, and everyone else is in blue. So I hope it's apparent from this that the research that the BHF funds compares very well against research funded by others. On this, we have research councils, we have the National Institutes for Health Research, and indeed the Wellcome Trust itself, and different types of research, fundamental science and translational science, including clinical studies. And in both categories, the British Heart Foundation comes out top. So I hope this gives you confidence that your money is used to a great effect. Now, the coronavirus pandemic has affected all of us, has affected all aspects of our lives. And the BHF and BHF-funded researchers have not been immune from that, unfortunately. So during the first wave in particular, when all research essentially had to stop, we took action to protect that very strong infrastructure and human capital that has taken 60 years to establish in the fight against cardiovascular disease. So we moved to continue funding our researchers, even though they were unable to do their research through no fault of their own. We supported them to join the national effort to fight the pandemic in multiple ways. Our clinicians actually rejoined the NHS to help with patient care. Many of our scientists joined the testing effort to support the national effort. Our medical director, Professor Sir Nilesh Samani, was really quite visionary in getting together with the leadership of the National Institutes for Health Research to establish a program, a flagship program, to address the urgent research questions impacting on people with or at risk of cardiovascular disease during the pandemic. We established an emergency scheme to provide time extensions our research projects based on need, and recognizing that our younger scientists were particularly impacted, we established emergency studentship extensions for the most severely affected PhD students. And on top of all that, we also made the strategic decision to maintain our competitive research funding schemes in order to protect the pipeline of talent and discovery. However, there is no hiding from the fact that the coronavirus pandemic has had a devastating impact on BHF fundraising income, and as a direct consequence of that, on our research funding capacity, as Charmaine has mentioned already. And this really does put at risk the pace of progress towards new and better ways of preventing, detecting, and treating heart and circulatory diseases, as well as a generation of future leaders in cardiovascular science and medicine. So to counter this, we definitely need your ongoing support, but we are also taking other actions. We are working with other medical research charities towards securing government match funding through a scheme that we've termed the Life Sciences Charity Partnership Fund. And we call on your support again on this also, to contact your MPs and ask them to support us in this approach to the government. Now, I want to end my presentation on a positive note by talking about a very exciting area of cardiovascular research, which has immense potential. But before I do that, I think I have another poll question that I need to put to you. Now, I probably, oh, you can see it. I was going to say I may need to come out of my slides, but hopefully you can see it. So the question is, how many people in the United Kingdom are living with heart failure? So lots of answers coming in. Fantastic, 83% voted. So the most popular answer is A, 920,000, if I'm reading the results correctly. Is that right? Okay. Are we allowed to give the correct answer, or does that come later? Well done. I think 51% of you got that number right. It's really sobering. There are nearly 1 million people who live with this devastating condition called heart failure in the UK. And this is really exactly what we are looking to reverse with the topic of today's research, which is healing damaged hearts. So healing damaged hearts, this field is also sometimes referred to as regenerative medicine. And the objective of research here is to use new technologies such as stem cells and tissue engineering, gene therapy, these complex sounding terms on this slide, which Sanjay is going to go into in a bit more detail and explain to us. But the objective really, the need for research in this area arises from the fact that unlike some other organs like our livers, our hearts have very limited capacity to repair themselves following injury and damage. For example, following a heart attack, if a large amount of heart muscle is damaged, our hearts cannot repair themselves. So the ultimate aim here is to use these emerging new technologies to repair the heart damage, to restore normal heart function, and thereby to maintain the quality and, of course, the quantity of life of patients with damaged hearts. Now, our researchers are already making quite amazing progress in this area. We have a BHF-funded professor at Queen Mary University of London who's at late stages of testing repair patches, if you like, that can be stuck on the damaged heart. And we have a professor at King's College London who's shown for the first time in a pig that it is actually possible to wake up the self-repairing capacity of the heart. But I don't want to spend any more time telling you about regenerative medicine because we have one of the world experts in this area joining us today. Our guest speaker, Sanjay Sinha from the University of Cambridge. I'm proud to say that Sanjay is a British Heart Foundation Senior Clinical Research Fellow, and he's also a consultant cardiologist in Cambridge. And I'm going to hand over to Sanjay to do the rest of the talking and tell us about his fantastic work. So Sanjay, over to you.
Metin, thank you very much for that introduction. I'm really honored to be asked to speak at this live and ticking event. And I'm delighted to present the work that we do in the lab, and particularly because we've been very heavily supported by the British Foundation. So a lot of the work that you're going to see has been funded by the British Heart Foundation. So I'm going to share my screen because I think the pictures and images of the research will really help to convey what I want to share with you. Now, okay, if I can get a thumbs up, that's okay, great. So I'm going to talk about mending broken hearts and blood vessels. And what I want to do is to give you a whistle-stop tour of what we do in my lab, looking at stem cells and how they can help us to understand and treat heart disease and blood vessel disease. And I'm going to start by telling you what stem cells are. Then I'm going to tell you how we can use this to do disease in a dish type work, type modeling, how we can then use it to repair damaged hearts. And finally, I'll touch on the work we're doing on how we can use stem cells to understand how COVID might affect the heart as well. So let's get straight into that then. What is a stem cell? So a stem cell, simply put, is a cell that can reproduce itself, so it can divide and make more of itself, and it can also turn into other cell types, what we call differentiation. So that's the essence of what a stem cell is. And we all have lots of stem cells in our bodies. So for example, the one that perhaps people know the best is the bone marrow stem cell. And so the bone marrow can generate all of the components of blood, red cells, white cells, and platelets. We have stem cells in our skin. They generate hair follicles, and in fact, they also generate skin as well. So the skin repairs and regenerates itself. There are stem cells in the gut. Many organs in our bodies have stem cells, but these are all adult stem cells. What they have in common is they're quite restricted in what they can generate. So the bone marrow can only generate blood cells. The hair follicle stem cells can only generate a hair follicle, and so on. The kind of stem cells, there's a different kind of stem cells that we work with, and they have much more ability to form different cell types. So we work with embryonic stem cells. Now, these stem cells are originally isolated from a fertilized egg when it's a ball of cells. And we can actually culture this fertilized egg, this ball of cells, and these cells can be cultured indefinitely in tissue culture. And what's special about these cells, these embryonic stem cells, is that if you give them the right signals, they have the potential to form any cell type or any tissue in the body. So they're very powerful. There's a very similar stem cell that we also use, and that's called an induced pluripotent stem cell. It's quite an involved title, but basically these were discovered by this chap, Yamanaka, in 2006, 2007. He won the Nobel Prize for his discovery a few years later. And the beauty of this discovery is that we can generate these cells that behave like embryonic stem cells from anybody. So what we do is we can take a piece of skin or even a blood sample from a patient, and we use just 4 of these proteins. These are little molecular switches that turn on different genes in these cells and turn something like a skin cell or a blood cell into a stem cell. And these induced pluripotent stem cells, as they're called, do all the things that an embryonic stem cell can do. So they can turn into all the tissues in the body. And we can then use that for drug screening and disease mechanism studies. I'm going to show you an example of that, how we do that in my lab. So we're very interested in Marfan syndrome in my lab. This is somebody who suffers from Marfan syndrome, and they have a characteristic appearance. So there's overgrowth of their long bones, they have very long fingers. In fact, some people have wondered whether Abraham Lincoln also had Marfan syndrome, although I think that's a question that will never be answered. But as well as it affecting their bones, it can affect their eyes, their lungs. The patients who have Marfan syndrome particularly suffer from this thing we call aortic aneurysm. Now, the aorta is the main blood vessel in the body that comes out of the heart. It takes blood to the brain, the rest of the body. Usually it's the size of a hose pipe. But in patients who have Marfan syndrome, the aortic wall gets weakened, and this pipe, this blood vessel, starts to stretch out. It can be turns into as big as an apple or an orange. And the wall gets very thin and weak, and obviously that can then tear. And if that tears, that's clearly catastrophic for the patient. And the problem is that we don't really know how to treat this. Yes, a surgeon can actually cut that weakened part of the blood vessel out and replace it, but it's a huge operation, and the bit that you don't replace can still continue to get bigger. So we don't have any definitive treatments that can stop it growing. Now, what causes it? Well, Marfan syndrome is a genetic disease. So there's a mutation or an abnormality in the DNA, and that can be passed down families. It occurs in about 1 in 5,000 people. And the mutation is in a gene called fibrillin-1. Fibrillin-1 makes an extracellular matrix protein. What's that? Well, if you think about the cells in the body as bricks in a wall, and the extracellular matrix is the cement or the mortar that holds the bricks together. Now, if there's a fault in the matrix or there's a fault in the cement because of a mutation in fibrillin, let's say, you can imagine that brick wall is not going to be very strong. And that's the case in these patients who have Marfan syndrome, the aortic wall is not strong enough. So what can we do? Well, we made a disease in a dish model of this. So we took skin samples, patients with Marfan's generously donated skin, very tiny samples of skin, actually just a few millimeters, and using that and the 4 molecular switches that I described, we made stem cells that were specific to those patients. And then we coaxed these stem cells into making smooth muscle cells. These are the cells that make up the wall of the blood vessel. And the power of this technique is that since these patients have an abnormality in their DNA, that's reflected in their skin cells. And so our stem cells and the smooth muscle cells we make from them all have the same genetic abnormality. So we then look at these cells under a microscope. I'm going to show you some images from an electron microscope. Now this has a really high magnification view of the cells and the matrix. Now most of you won't be experts on looking at these pictures, but have a look at it anyway. What I'm showing you on the top right is a picture from a normal smooth muscle cell that we've cultured in the lab. At the very top and the bottom are cells. In the middle, that gray area you can see, the filling in the sandwich, is the matrix. Normally, it looks like that. It's compact, nice and smooth. The picture below it is smooth muscle cells from a Marfan patient that we've made using this model system in our lab. On the left part of the picture is a smooth muscle cell. In the middle, you can see the matrix, or the cement, if you will. And you can see how abnormal it is. It's chaotic, it's broken up. And really, that's what we see in patients' aortas, that's what we see in our dish. And that you can see why a patient's aorta might not be very strong if that's what it looks like. We can recreate all of the differences, all of the abnormalities in a patient's aorta in our tissue culture dish, and that's why we call this a disease in a dish model. Now this is really powerful because we've got a disease in a dish. Now we can say, well, how does that mutation in the gene cause this abnormality we're seeing under the microscope? Even more importantly, from the patient's point of view, we can use this to screen for drugs that might prevent that abnormality that you're seeing. Now, doing drug tests in patients is very difficult. But with this sort of disease in a dish approach, I can test hundreds. In fact, we've tested thousands of drugs on this disease in a dish models at different doses in different combinations. And we found new drugs that can prevent the abnormality in the dish. This is really exciting. We are now using these in our models and will anticipate going on to do clinical trials with some of these drugs once we've identified really the best combination and the best candidates. Okay, I hope that gives you some idea of the power of this disease in a dish approach. I'm going to go on now to a different aspect of our work using stem cells. So this is a question for the audience. Which one of these is the odd one out? Now, usually I get audience to write back, sort of to yell out, but actually in Zoom is slightly different. So I'll give you the answer. The odd one out is this unfortunate chap clutching his chest. And the question is, why is that the odd one out? Well, Metin alluded to this earlier. Humans can't repair damage to their hearts. The others, you've got zebrafish, newts, got mice when they're first born, newborn mice. They can all repair damage to their heart. If you give them a heart attack or cut a piece of heart away, they'll repair that. But adult humans can't do that. And so, of course, what happens is that after a heart attack, we can't repair the damage. This is an example of somebody who had a very large heart attack and unfortunately didn't survive. This is a section through their heart, and you can see this dark, black, red area where the heart attack was. That's necrotic muscle, hemorrhagic, and that's all dead tissue. You can also see, actually, there's a white area in the middle where this person had a heart attack previously that they did survive, and that white area is scarring, where they've got a scar. That's not muscle, that's just scar. And you can see that if this person had survived their heart attack, they would have had very little normal muscle left. And so the heart would have been weak, and undoubtedly they would have had heart failure. I put a very big number there at the bottom. That's 1 billion, 1,000 million. Why have I put that number? Well, that's how many heart muscle cells that we lose, or somebody who has a reasonably big heart attack would lose. That's how many heart muscle cells die. So that's the challenge. If we're going to regenerate that heart, we have to replace 1 billion heart muscle cells. It's a huge number. There's only about 3 billion in the whole heart altogether. So, and most of the treatments that we have, there's lots of great drugs that improve the symptoms and the outlook for patients with heart failure, but none of them tackle this problem where the patient might have lost 1 billion cells. And what does that do? What does somebody with heart failure, what does it feel like? Well, I borrowed this from an advert from the British Heart Foundation. You may have seen this actually. When the heart doesn't work properly, blood pools in the lungs, and you can even get fluid building up within the lungs. Patients sometimes say that it's like being underwater or drowning. So it's a very graphic example of what having heart failure actually feels like. So how can we treat this? If drugs can't do the trick, how to replace this? Well, stem cells and regenerative medicine offer us an opportunity here. So we can use stem cells to make heart muscle cells. I've shown you how my lab has made smooth muscle cells. Well, we can do the same, slightly different signals to coax the stem cells to make heart muscle cells in a dish. What does that look like? Well, if you take a look at normal cells, these are just some skin cells. If you look under the microscope, this is what you see. They don't do very much. Most cells will just sit there. If you make heart muscle cells and have a look at those under the microscope, this is what they do. So this is a sheet of hundreds of heart muscle cells, all joined together, all beating together. And this is just looking down a microscope. This is exactly what you'll see if you come to my lab, and you can see this. It's really quite amazing. Every time I see this, I'm amazed. So can we use these heart muscle cells that we make in the lab to try and repair or regenerate that damaged heart? So we worked in animal models. We used a rat model where we gave the rat a heart attack, and then 4 days after that, we injected these heart muscle cells into the damaged area. And you can see in the rat heart, this is 3 months after we've injected these heart cells. These are human heart muscle cells, and you can see the human cells in bright green. The dotted line shows the area of muscle that was damaged, the area of heart attack. And you can see the human cells have really repopulated that quite nicely. They integrate with the rat heart muscle cells. What they do is they beat and they improve the function of that damaged rat heart. And that continues for over 3 months. They bring in new blood vessels and really form quite a nice repair. And what we showed is that just heart muscle cells by themselves aren't good enough. We get a much better result if we combine them with this other cell type we call epicardium. And these are supportive cells, and they help the heart cells to do their job. So you need a combination of cell types to get the best result. Okay, just injecting cells into a damaged heart, although we're getting quite nice results, about 90% of the cells we inject there get washed away. And since we need to replace 1 billion heart muscle cells in a human, I mean, a rat heart is what, the size of my thumb, a human heart's the size of my fist. So we need a lot more cells for a human heart. We don't want to lose 90% of them. How can we do this in a better sort of way? So we're making heart patches. The heart patches we're making are a special collagen scaffold. They have special handholds for the cells to sort of attach to. And then we make these sorts of patches. This is a patch that's probably the size of my fingernail. And again, it just contracts away nicely in a dish. So we can then just stitch that onto a damaged heart. We're doing those studies again in animal models to show if we can stitch it onto a damaged heart, whether it can repair and help that heart to beat more strongly. So that's the sort of work we're currently pursuing in terms of heart regeneration. I think there's a lot of promise there. I want to touch briefly on COVID for sort of my final example. We all know that this virus, the SARS-CoV-2 virus, can attack the lungs, and that's the principal area attacked. But there's lots of reports that it can also cause heart damage. You may have seen headlines such as this one in the Daily Mail, that COVID-19 can damage hearts in half the patients that this study looked at. That might be overstating a little bit because this was in patients who all had heart scans. But certainly patients who come into hospital, maybe 1 in 8 or 1 in 10 of them, also have heart damage. And the question is, how does that happen and can we prevent it? So the virus can either directly affect the heart, and we can look at that using our model in a dish. So we can put the virus onto the heart cells. These are human heart cells in a dish and see exactly what happens. And that's what we've been doing. We can also look at the patient's serum. So we can take the blood, the serum is the part without the clotting factors, and say, is there something in there that might actually be causing the damage? So it might not be a direct effect of the virus on the heart muscle. It might be something, cytokines, inflammatory factors, that are in the patient's blood that actually causes the damage. So we've been doing some work looking at both of these possibilities, see what might be causing damage, and trying to then see whether there's something that we can use, at least in a dish, to block that damage to heart cells, which might then be used, potentially be used in patients. So that's just briefly touching on the work we're doing with COVID. I want to end just by talking about the British Heart Foundation and their tremendous support for stem cell research. I'm sure that as supporter of the British Heart Foundation, you're probably familiar with this advertising campaign that the British Heart Foundation had a few years back on the Mending Broken Hearts appeal. And that was use this zebrafish again, because obviously the zebrafish can repair its heart, as the sort of mascot for this appeal, if you will. And this appeal, one of the things it did was to set up regenerative medicine centres around the country. So my group is part of the Oxford-Cambridge Centre. There's centres in Imperial, also in Edinburgh. And these were established in 2013 and then renewed in 2017. And these have been absolutely tremendous for supporting regenerative medicine around the country, as well as in my own lab. This is a picture of my lab. They're a tremendously talented bunch of scientists, PhD students, and so on. And as you can see, really very enthusiastic about their work. But just to give you an idea of how important British Heart Foundation support is, if we take away the people who are supported, where their salaries are supported by the British Heart Foundation, and where the work they do in the lab is supported, let me start taking away the people who are supported fully by the British Heart Foundation. So you can see that's quite an impact, and you can see perhaps how important the work that you do in raising funds for the British Heart Foundation is for the work that we do. And this is just a representative, I think, of hundreds of research labs around the country that rely on BHF support. So I do want to say how sincerely grateful we are for the work that you all do in raising funds for the British Heart Foundation and restore my lab to its glory. What we want to say really is thank you to British Heart Foundation, but particularly the funders, the people who back it, the people like you. Thank you very much. I'll end there, but happy to then take part in any questions. Thank you.
Lovely, and thank you, Sanjay. I know that everybody on the call today will join me in thanking yourself for such an amazing presentation and giving us such insight into the power and potential of regenerative medicine to help people, particularly with heart failure, and hearing you and Metin describe just how many people, nearly 1 million people of the UK, are living with that today. We get a sense of the scale of that, and many people, of course, on the line might actually suffer from it too. So I know that would be incredibly hopeful to us. So huge thanks to you. Now we've got about 10 minutes, 15 minutes or so for questions before we let you get on with your evening as well. And we love hearing your questions. We've already had plenty submitted in advance that I'm going to ask our moderator, Christy, to talk us through. But if there is something that strikes you whilst we're speaking, please do add it in the chat box, and we commit that we will definitely get back to you, if not today, then in follow-up. So Christy, if I can ask you to start us off with questions.
Of course. So our first question is to you, Charmaine, and it is, I know the BHF and others are lobbying the government to support research at risk because of the pandemic. Can you tell us how that work is going?
So great question. Thank you to whoever asked it. And so this, just to recap, is a really important piece of influencing we're doing to try and protect the funding available for vital medical research Just to give you a sense of how that's impacting the BHF, that the pandemic has halved our net income this year, which means our budget for amazing and life-saving research, like you've heard Sanjay describe today, has dropped from over £100 million to £50 million this year, and it's going to take many years to recover. So we're really proud, and as Chief Executive of the BHF, I'm really proud to be standing in solidarity with 150 other medical research charities, urging government to set up a life sciences charity partnership fund to help match fund research funding from charities through the next couple of years in order to protect both the amazing projects and programs you've heard some of today, but also the amazing talent. So Sanjay showed pictures of his labs. We're really proud to be funding hundreds and hundreds of PhD students and postdocs around the UK. And we're particularly worried about not being able to keep the funding for our young talent and our career pipeline supported. So the one thing that you could do to help us today, if you have a few moments, is to actually join our campaign to email your MP to ask them to back the call for support for this fund. And I know that Christy and colleagues will put the link in the side chat. So if you do get a couple of minutes over your cup of tea once we've finished, please do take a moment to click and help us make a difference. Thank you.
Thanks, Charmaine. Our second question is for Metin today, and it is, given the fact that we know charities will be facing the effects of this pandemic for years to come, what is the BHF's priority in terms of research? Are there some fields of research that may suffer more than others?
Thank you, Christy. What an insightful question, and a very important one too. I'm going to start by saying that we take the responsibility of investing the money that you entrust in us very seriously indeed. So it's always been the highest priority for us to select the best people, and as I said in my presentation, the brightest minds wherever they may be, to do the research that has the greatest potential for impacting on cardiovascular health, for helping patients with or at risk of cardiovascular disease. So we have had to make some very difficult decisions in the light of the financial impact, as Charmaine has mentioned. Pretty much overnight, our research budget went down from £100 million to £50 million. But we made the strategic decision that the priority has to be to fund the very best applications, the very best people. So we've always been very stringent in how we invest your money. In the normal world, pre-pandemic world, we probably received 3 or 4 times more applications than we could afford to fund. We're still getting those applications, many of which are quite brilliant, but we are funding fewer of them inevitably because the budget has shrunk so much. So we're not excluding any type of research because there are so many different types of heart and circulatory diseases. We are focusing our funding and on our support to the very best of those applications that we receive, those that have the greatest impact for benefit for patients.
Great, thank you, Metin. Our next question is for Sanjay, quite a big one. What do you think the next big breakthrough will be in the field of regenerative medicine?
Yeah, this is a great one to think about, isn't it? I think the next big breakthrough, I think it's sort of about to happen pretty soon, will be cells that don't cause an immune response. One of the biggest challenges is putting cells into patients. The patient's own immune system tends to attack the foreign cells. So what we need is cells that don't trigger that immune response. So what people are working on are stem cells. We call them hypoimmunogenic. They don't trigger an immune response, but they still have to be safe. And those cells would then be used to make heart muscle tissues and so on. And actually for other areas of regenerative medicine too, whether it's Parkinson's disease, whether it's diabetes, having that kind of hypoimmunogenic cell that can then be given to all patients without trying to match them immunologically, without having them to be on immunosuppressants, will be a huge breakthrough. So I think that's the next one, and I think we're going to see that in the next few years.
Great. And a follow-on question for you, Sanjay, around your disease and dish model. So can your Marfan disease and dish model be used to replicate other kinds of diseases in the lab, such as coronary heart disease or vascular dementia?
Yeah, so we've got a model of vascular dementia. So there are genes that cause vascular dementia, things like CADASIL and so on, and we've got models of that. But what we're talking about really, what the question is asking is diseases like vascular dementia or heart disease that are not caused by these single gene mutations, these quite rare disorders. And actually what we're doing is studying the rare ones because they give us insight into the more common diseases. So vascular dementia that's not caused by a genetic condition, or coronary heart disease that's not caused by a single gene. So yes, we can do disease modeling, and we're starting to do that as well. It's more complicated, but we can do that.
Great, thank you. And another question for you, Sanjay. Is there any evidence that COVID-19 is causing long-term damage to the heart? And I know that you've touched on this. So if you could just expand on the damage that occurs, particularly to those with underlying conditions.
Yeah, this is it's a really important question right now, and we have some information, but we're in a state of sort of imperfect knowledge because we don't have a long enough follow-up. We've only followed patients up for a small number of months so far. It does seem that in some patients, and particularly those who are badly affected in hospital, that there are long-term consequences. Even when they get better, that there may still be changes in the heart. We don't know exactly how many patients that affects, though, and to what extent this is going to be a problem. So this is a bit of an area that certainly, yes, there is a long-term effect. We don't know how big an issue that's going to be. So I think that is going to need a bit more study. But there are lots of studies following groups of patients out, doing MRI scans and so on, to see what the long-term consequences of getting COVID are. So I don't have a full answer, but there is a long-term risk potentially there.
Thanks, Sanjay. We've had a lovely question through from Philip Reed. Thank you for joining us, Philip. This one's for Charmaine. Philip says, I'm currently waiting for treatment as I have a leaky mitral valve and I'm 43. I'd like to look at what options there are for me to help the BHF. And also help you moving forwards. I'd love to share my experience and help to educate and support others. How can I look to do this?
Oh, well, firstly, thank you for offering to support us. And I hope you're well and taken care as well at this time. The short answer is there's loads of ways you can get involved depending on how you want to. And probably the simplest thing is have a quick look at our website. But please also do get in touch. I know we've got your details from this call today. So we have a brilliant team who work with people who've been affected by heart and circulatory disease to help share their stories, to either help raise support for the BHF, or indeed to help raise awareness of important things like the support that we offer for patients as well. So thank you for your kind offer. I know the team will be in touch as follow-up to your kind offer. And please do check out our website for ways in which you can help support us. As you heard me say in the opener, people are doing so many remarkable things to support us. So people are doing lots of their own events, whether it be swimming in rivers or virtual bike rides. But there are other simple ways in which you can support the BHF, either through simple donations. People give us incredible gifts in legacies and their wills, and in other ways. So in whatever way you can, whether it be donating stock or indeed money or just your time, we very much appreciate it. And please have a look at our website and let us know. Thank you.
Thank you, Charmaine. We now have a question for Metin related to the statistic that you showed during your presentation. Why do so many people have failing hearts in the UK?
Thank you, Christy. Another very good question. I mean, as I said during my talk, that is a sobering number, nearly 1 million people. But I don't think the United Kingdom is doing any worse than any other country. In some ways, the success that the medical field, the cardiology field, has had in treating heart attacks, so that more and more people actually survive the acute event, the acute heart attack, is potentially also contributing to this large number of heart failure patients. As patients get older with damaged hearts and they have other risk factors, they're more prone to develop heart failure. So what's the answer? And I think the answer is twofold, and we're trying to make progress with both. One is to find better ways of treating heart failure, and that's what a lot of our research is about, including what we've heard today. And at the other end of the spectrum is making sure that patients with heart failure in the United Kingdom have access to the best existing treatment for heart failure. And we have a very active healthcare innovation team that's working very hard to encourage the adoption of the very best treatments in the NHS and the availability of that treatment to patients with heart failure.
Thank you, Metin. And we have one more question for Charmaine before we will close. What tangible things can supporters do to help the BHF weather the COVID-19 storm we face today? You're on mute, Charmaine.
Forgive me, everyone. You'd think I'd found the mute button by now, wouldn't you? So my apologies. Again, thank you. I can see in the questions that are coming through, there are lots of people offering to support, and everything we simply couldn't do what we do without you. The work that Sanjay's described, the hundreds of projects we have active across the 4 nations in the UK, and the thousands of researchers we support, we simply couldn't do that without your support. There are lots of ways you can get involved and support the BHF. As I said, the quickest way to have a look around at your choices is have a look at our website, which is at bhf.org.uk. And whether you want to do your own thing and raise funds at home, or do activities and sponsored activities, as so many people are doing brilliantly, or you just want to simply donate, or indeed consider other ways to support or do a declutter, all of them make a huge, huge difference. So I would just say a huge thank you to you again for joining us today, but also offering support so freely as well. It makes a huge difference. So before drawing us to a close, I'm just going to say again, a huge thanks to Sanjay for giving us such an articulate presentation and making so simple the complex research that he and his team are brilliantly doing to look at the potential for regenerative medicine, and particularly reflect on the hope that that gives for people with heart failure, for whom this is clearly a vital programme of work. So I know that you will be there virtually joining me and offering my thanks to Sanjay for being so eloquent. And I can see on the comments in the chat box as well that people really love the way you brought that to life, Sanjay, so huge thanks. And a particular thanks also to Metin for giving us a snapshot of the work that the BHF does. I wonder if I could ask you to answer a couple of questions before we wrap up. So let me ask question 4 to you, our 4th question, which is, at the start we asked you how much you felt you knew about research into healing hearts on a scale of 1 to 5, with 5 being the highest and 1 being the lowest. How are you feeling now? See the numbers come in. It's looking good, people. We've learned something today. I can tell I've learned things today, my goodness. Okay, lovely. And literally hundreds of people still on the call, so thank you so much for being with us this afternoon. It's brilliant to have you with us. We've got a couple more people. Here we go, look at that. Oh, fantastic. Sanjay and Metin, look what you've helped us do. So we've gone from a position where we thought that probably we're a 2 out of 5, and now after just under an hour, we feel as a collective that we're around 4 out of 5, 50% of us, half of us feel we're pretty confident and know a little bit more about research into healing hearts. So thank you for that, Sanjay and Metin, and helping us get there. I'm going to ask you one more question before we wrap up, which is, having joined this event, and we really love the live and ticking events and spending time with you, do you feel this has increased your likelihood of supporting the BHF and the research that we do? Thank you. See lots of numbers coming through. Fantastic. Thank you very much for sharing that with us. That just gives us some useful feedback to think about what matters to you and how to organise these events as well. So thank you very much for that. So I we just like to finish off by saying, again, huge thanks to you, our supporters. As BHF's Chief Executive, I could not be prouder of our BHF team, our colleagues, our volunteers and our supporters. Our BHF family is really helping to protect our life-saving research through this unprecedented challenge. And everything you do to support us makes a huge difference, whether it be donating, emailing your MP, decluttering, everything makes a big difference. So huge thanks. We know these events are really exciting for us, and I think they are for you too. We're hearing lots of great feedback, but please do take a moment to give us a bit more feedback when we email you a link to our survey later on today. Everything you tell us helps us shape these events for the future, so please do that. And the next one of these lovely events, our Live and Ticking events, is on the 16th of December, just before the festive season. So if you'd like to join us at the same time, we'd love to see you, and again, love to be answering your questions. And if you want to sign up your friends and family to join us too, they can join us and sign up at bhf.org.uk/public events. So super easy to do. And lastly, you will get a chance, if you really want to hear Sanjay and Metin do their thing again, to rewatch this, which will be published and is recorded. Today's session will be published next week on our website. So it'll be an opportunity to rewind and also to see some of the slides that I know that people have requested as well. So without further ado, I'm just going to say a huge thank you again, particularly to Sanjay and Metin, but a huge thank you to you for everything you do to support the BHF and for joining us today. And until next time, take care. Thank you.